And let me remind you that on April 20th, that'll be Tuesday, I think next Tuesday, I'm going to be at jury duty, and so I'm going to pre-record the lab, and we'll have the lab at 5 o'clock on April 20th. And I'm hoping to be back home in time for the lecture. And so the lecture will be at 6:30. And hopefully this will be the only day that this happens, but if I do get selected for jury duty, this schedule may continue until I get off of jury duty. And I will let you know as soon as I know further about that. Any questions on that? All right. You should be seeing my screen. Let's get rid of this. I'll leave that up, but I won't use it. One problem with my new system is I can't plug in my thumb drive, but I've got stuff here. Oh, I wanted to mention there's a couple of extra credit assignments on the web. So if you haven't checked that out, please check that out. I'll try and quickly get there. Most of you have already sent in the picture, but there's, I think, 3 extra credit points if you send in a picture. Starting this week, there's the extra credit, is it study guide? Is that what I call it? All right, let me check and see what I call it. What you do is you get together with somebody. You can use Zoom, you can use your phone, and just study over some material that will be tested on. Let's go to week 2. Extra credit study guide. So it's the extra credit study. And what you do is you have to study for at least 30 minutes. And then send to the Canvas page, which will be the extra credit study 2. And you'll have a link up here for downloading. Who was in your group and what you studied, when you studied. You have to study for at least a half an hour over some material that is testable. And you'll get 1 extra credit point for getting together and studying. Like I said, you have to turn in the full names of everyone in your study group, and that's so that only one person needs to turn this in your study group. So make sure you have everybody in your group. And I need the full name so I can get them the credit. Tell me when you studied and what you studied. Then submit it. You can type it in directly, or you can put it in a document and then submit it. Any questions about that? That has to be done by 11:59 p.m. by April 17th. That'll be this Saturday. Can you, let me put the volume up a little louder. Can you repeat that?
Should only one of us submit the paper then?
I still didn't catch that. Can you say that again?
Should only one of us submit the paper, or should everyone submit their own documentation?
Oh, yeah. Just one person in the group needs to submit the document. As long as you have all the names, I can get everybody in the group the credit. Okay? And if you don't know how to get a hold of other students in the class, you can click on the people link and then email people when you want to try and get together with someone to study. Usually people get into different groups and then they continue studying at a specific time each week. Let's see if there's anything else I wanted to talk to you about. I think there's an extra credit quiz. Let me get this out of the way. I already mentioned the extra credit picture. Yeah, practice quiz. I think there's an A and a B. Yeah, there it is there. Practice quiz 100A. This is questions that are short answer or essay questions. And if you submit that, you'll get something like 1 extra credit point. And then you can take the practice quiz. And this one will be the multiple choice questions. You have to use Respondus Lockdown Browser for doing this. I highly recommend you getting Respondus working on your system, downloading and trying it out. In case you're one of the few students, and usually one student every other term has trouble getting Respondus to work on their system. And if you have trouble getting it to work on your system, call the, whatever it's called, the computer help desk or the computer IT system, and they're very good at getting Respondus working on your system. If you submit this by April 14th at 11:59 p.m., you'll get, I think it's 0.1 extra credit point per question, and there's 10 or so questions, and so you'll get about 1 extra credit point for submitting this. And both of these quizzes are good because there'll be quiz questions similar to what you'll see in the quizzes in my class. And that's why there's 2 quizzes, an essay quiz and then a multiple choice quiz. Now, none of these questions will be on your first quiz, but there will be some similar material. Any questions about any of that? All right. And then remember, you have a worksheet for lecture 2 that is due this Saturday. And then there's one, actually there's 2, I think, for the lab, because we have 2 labs this week. And also, I corrected the worksheets for Lab 00 and Lab 01. And you can, if you didn't get full credit on that, you can resubmit your answers. And that'll be for full credit. That has to be done by, I think it's 11:59 p.m. on Thursday. And the same with worksheet lecture 1. If you didn't get full credit on that, you can resubmit your answers by Thursday. Let me look on that to make sure. That I changed the date there. Yep, you have until April 15th. All right, any questions about any of that? If not, let's take a look at what we're going to be doing today. I already know we're going to be covering Chapter 1 and Chapter 10. Hopefully we'll finish that today and get started on Chapter 2. We'll see. Went a little too far. Yeah. Okay, that's what we're doing. And then the practice quiz is due Wednesday. I think I'll leave that up. I'll use it for the lab. I had multiple 2 here's in this document, so I'm not entirely sure. Did we talk about this slide? I think we did talk about Louis Pasteur's experiment.
Yeah,
I'm
we
not
are.
hearing anyone. Is anyone there? Hello?
We were past there, yeah.
Okay. Um, so beginning with Pasteur's work, there were a number of discoveries made in microbiology, including the relationship between microbes and disease, and this gave rise to the germ theory of disease, that germs, meaning microbes, could cause disease. And out of this work came the work of immunity, that the immune system responds to defend the body, and also the work in antimicrobial drugs. And so we'll talk a little bit about each of these, especially the antimicrobial drugs. Let me put this on the bottom. In 1850, you don't need to know the date, Pasteur developed the basic concept of aseptic technique and then used it to grow his cultures so they wouldn't be contaminated. Robert Koch in 1881, and I'm not pronouncing this correctly because that's a German name and the English don't have the pronunciation for this CH. There's a German sound. If anyone speaks German, you know what I'm talking about. He developed the technique for streaking for isolation on solid media. This was important because it gave an easy way to get a pure culture. Louis Pasteur had a way for getting a pure culture, but it wasn't easy to do, and we'll talk a little bit more about that in the lab. And then shortly after, Robert Koch did the streaking on a solid media to get isolated colonies. Someone in his lab, an associate of his, developed nutrient agar, a very good general purpose media for growing microbial growth. And about the same time, Sergei Winogradsky developed the idea that microorganisms were involved in the biogeochemical cycles, such as the nitrogen cycle, recycling nitrogen in the Earth's ecosystem. And Winogradsky also studied the sulfur cycle at the same time. Shortly thereafter, Louis Pasteur in 1857 showed that microbes are responsible for fermentation, and he showed that microbes can create alcoholic fermentation, converting sugar to alcohol, and that is how beer and wine get made. He also showed that microbial growth is responsible for the spoilage of food, and he was specifically looking at beer and wine, and that microorganisms could grow in the beer and wine and metabolize the alcohol and make acetic acid, and that would spoil the wine, fermenting it to vinegar. Any questions about any of that? Now, obviously, we do use that for making vinegar, but if you're trying to make beer or wine, and this happens, it spoils the beer or the wine. The taste is off and it's a sour. Out of this work came Pasteur's work, where the spoilage bacteria could be killed by heat, and that he could treat his beer and wine with the temperature hot enough to prevent the growth or kill the growth of the spoilage bacteria, but not hot enough to evaporate the beer or the wine. And this led to the process of pasteurization, the application of a high heat for a short time. It's used in a variety of foods. It's still used for beer, which Louis Pasteur first used it on beer and wine. It's very seldom used for wine, because pasteurization does cause the wine or the alcohol in the wine to evaporate, at least some of it. So it's not used so much in wine, but it is used in beer And it's very commonly used, of course, in dairy products. Like almost all milk is pasteurized. The studies on fermentation, that microbes can affect an organic material, leading to a change in the organic material, meaning converting the sugars into alcohol in beer and wine. It led to the question, could microbes have a similar relationship with plants and animals? Might microorganisms grow on an animal and then cause disease? This idea came to be known as the germ theory of disease, and it was an important concept because originally people believed that poisonous vapors were responsible for causing infectious diseases. For example, if someone got cholera and they were walking through the swamp, they thought at the time that it was because the person breathed in the poisonous vapors in the swamp. They didn't know that there were microorganisms in the swamp that could also give cholera. And that led to the germ theory of disease. I'll let you read over this. I'm not going to talk about that. We will talk that Robert Koch then went on to prove the germ theory of disease when he found the bacterium Bacillus anthracis caused the disease for anthrax. And that was the first case where a given bacterium was shown to cause a given disease. And then Robert Koch wrote up the experimental steps he used for showing or proving that a specific microbe causes a specific disease. And these steps are called Koch's postulates, the steps you use to show that a given organism causes a specific disease. Now, understand that Robert Koch did not propose the germ theory of disease. He was the person who proved it, or the first person who proved it. Any question about that? Actually, the germ theory of disease grew up over time by many people. It actually was Virchow who first wrote it up, but like I said, it was a concept that was developed by many people. And then Robert Koch proved it. And then in 1884, you don't need to know the year, he postulated his steps for showing that a given disease is caused by a specific microorganism, which we call Koch's postulates. And we'll talk about Koch's postulates about 3 times in this class. So that's one of the important concepts you need to know throughout the term. And you'll be tested on it multiple times, at least in the first quiz and certainly in the final. And there's one other time you'll be tested on it too. And you'll know because we'll talk about Koch's postulates. Now, vaccination was another concept that came out of the early microbiology, but actually vaccination started before microbiology. And the first vaccine was developed in 1796 by Edward Jenner. He made the first vaccine against smallpox, and what he did was he inoculated a person with the cowpox virus, and then that person was protected against smallpox. It was not understood at the time, in nearly 400 years, why Edward Jenner's vaccine was working. And how the understanding came about was the work of Louis Pasteur, who went on to show that cowpox was a related virus to smallpox, and so when the immune system responded to cowpox, it at the same time responded to, or at least part of it responded to smallpox. And that is how the vaccine worked. So when you gave cowpox, you were all protected against cowpox, by the vaccine, and you were also protected against smallpox. Any question about any of that? Okay. Vaccination is a term derived from the word vacca, which is a word meaning cow, from cowpox. And the protection that you get from a vaccine is called immunity. The work on vaccines, which, like I said, Louis Pasteur really developed it and then went on to make a number of vaccines. I'm afraid I don't know all the vaccines he made. The first vaccine he worked with was actually one not for humans, but was for chickens. He was looking at a disease that was hitting the poultry industry, and he made a vaccine against some viral disease. I don't remember what it was. And he experimented with it and found out why vaccines work, and then went on to make several other vaccines. The only one I remember off the top of my head was he's the one who made the vaccine for rabies. And we'll talk briefly about that later in the term. Out of this work came the concept of chemotherapy, the treatment of disease with chemicals. A chemotherapeutic agent can be used to treat an infectious disease. And these can be synthetic drugs, ones that are man-made, or they can be antibiotics. And specifically speaking, the term antibiotic is a chemical produced by a bacteria or a fungi that inhibits or kills other microbes. Now that is the textbook definition for an antibiotic. And by this definition, penicillin G is an antibiotic. But by this textbook definition, carboxycillin and even ampicillin is not an antibiotic. That it would be a synthetic drug because they're man-made or they are like, sorry, I'm losing the name of it. I just said it too. Ampicillin is made by, I think, a fungi, and then it is chemically altered by humans, and so it's a semi-synthetic. And therefore, by this textbook definition, it's not an antibiotic. Now, that's not the way that most people and the way that most clinicians use the term antibiotic. And most people, when we're talking about any of the penicillins, we call them an antibiotic. And we use the term antibiotic to mean a drug that specifically works against bacteria, meaning an antibacterial chemotherapeutic agent. Any question about any of that? Chemotherapeutic agents or chemotherapy does not tend to be used this way in the general public. Usually when we're talking about chemotherapy, someone is referring to the treatment of a cancer, meaning a drug used to treat cancer. And you can use chemotherapeutic agent to mean treatment for really any disease. It doesn't have to be just cancer. Quinine was the first drug that was ever used to treat a disease, and they didn't quite use it as pure quinine. They first used it as the bark of the cinchona tree, which had quinine in it. And when you took this bark, it could be used to treat the disease malaria. And this was first used in Europe as early as the 1600s, something like 1650. Okay, any question about that? So that was the first drug that we ever had that was effective, I should say in the West, that was effective at treating a infectious disease. Paul Ehrlich then went on to find the first drug to treat syphilis in 1910, and he called these his magic bullet, something that could be used to treat an infectious disease. Any question about any of that? We'll talk a little bit about that. What he did was he just simply went through different chemicals and then tested them to see if they prevented syphilis. He was using it in rabbits. And he just went on from one chemical to the next. And after about 500 chemicals, he never got anything that worked. People in his lab put a sign over the door, "Abandon all hope, those who enter here, or ye who enter here." Because they thought it was a hopeless task, testing all of these chemicals. But shortly after 500 drugs were studied, they came across the first magic bullet, Salvarsan, a synthetic arsenic drug, which was successful in treating syphilis. And that happened in 1910. You don't need to know the date, but you should know that Ehrlich found the first drug that was capable of treating an infectious disease. And then shortly thereafter, in 1928, once again you don't need to know the year, Alexander Fleming discovered the first antibiotic. What he did was he streaked out his bacteria on a plate, and he was a sloppy microbiologist, and he got a contaminant growing on his plate. And then for some reason, he left the plate on the counter at room temperature and then went away for the weekend on a vacation for the weekend. And then when he came back to the lab, he looked at the plate that he had left on the counter and discovered that the bacteria was growing, but he had a contaminant on the plate. And he noticed around the contaminant, meaning around the Penicillium mold, there was something coming out of the mold that was preventing the growth of the bacteria. And he correctly concluded that something was coming out of the mold that prevented the bacteria from growing. And he called this penicillin. And then he did further research on penicillin. He never got it purified, but he did show that it was effective at stopping the growth of most bacteria that were gram-positive. He was not able to ever purify penicillin from his cultures, largely because he just didn't have the technique way back in 1928. So he never developed the first antibiotic that became clinically used. The first antibiotic that was clinically used was a sulfur drug, sulfonamide, which was developed in 1935, and it was a man-made drug, so there was no real need to purify it, and so it became the first clinically available drug. And then penicillin was purified in the 1940s and then became the second antibiotic that was clinically tested in people and mass-produced. Any questions about chemotherapy? So when we're talking about bacteria, you should realize that bacteria were once classified as plants, and hence that gave rise to the term flora for microbes. And if you ever hear me talking about microflora, I apologize, that's the old term, and it's a term that I was taught when I went through microbiology, but that was many years ago. The correct term is microbiota, and this term is supposed to replace microflora. And we use this term when we're talking about the normal microbiota, the microbes that are normally present in and on a person, or if we're talking about another species, on that species, and that could be a species of animals or plant. All animals and plants host symbiotic microbial communities that grow with that animal or plant. And that we call that animal or plant's normal microbiota. The normal microbiota are central to that animal or plant's welfare. So our normal microbiota helps keep us healthy, and it does a number of things for us. The normal microbiota is usually found on and in the skin, the oral and nasal cavities, the respiratory tract, almost everywhere all the way down to the lungs, the digestive tract, the urethra. So all of the skin, the outlaying mucosal membranes. It's not, the normal microbiota is usually not found internal to the body, such as in the bone, the tissues, the blood. The normal microbiota is helpful in preventing the growth of pathogens. They can compete for resources or for space, preventing the pathogen from getting established. They can secrete toxins that inhibit the growth of pathogens. And we'll talk a little bit about that later. Let's move on. They can produce nutrients and growth factors which are helpful for their host. For example, humans get some of their folic acid and vitamin K from their normal microbiota. And the rest you have to eat in your diet because we don't make folic acid or vitamin K. And the normal microbiota for all animals is important in helping in the animal's digestion, including humans. E. coli and other bacteria in our gut help us to digest our food. The normal microbiota do not normally cause disease, so normally they will not make you sick, but in special circumstances, they can cause disease. Any question about any of that? An infectious disease is when a pathogen overcomes the host resistance, starts growing in the host, and then causes disease. And what happens to that is, we'll talk about this later, the microbe starts doing damage to the host, and that gives the signs and the symptoms of disease. An infectious disease is caused by a pathogenic microbe. A pathogenic microbe is a microbe that can cause disease. Remember that most microbes do not cause disease. Only a small number, a small percentage of microorganisms can cause human disease. All right, we're going to briefly talk about emerging infectious disease, EIDs. This is a new or changing disease that is increasing or has the potential to increase in incidence. Right now, we're under a pandemic by an emerging infectious disease. What is that?
COVID-19.
Yeah, COVID-19 is an emerging infectious disease. Does anyone know another emerging infectious disease? Can you say that a little louder?
SARS?
Try again.
SARS?
SARS. Well, SARS, as far as we know, is extinct, So no, hopefully that is not an emerging infectious disease. If it ever comes back, it would be an emerging infectious disease. So in 2003, and I think it came back one other year. Does anyone remember when it came back? I don't remember when it came back. When it first came out in 2003, it was an emerging infectious disease. And hopefully it's now extinct. And boy, I hope the world never sees it again, but we'll see. Another emerging infectious disease, one that has the potential to increase in incidence, is bird flu. Hopefully that'll never spread among humans, because bird flu is a flu that is actually fairly toxic to humans. So I have a bunch of slides on emerging infectious diseases. All I'm going to hold you responsible for is what is an emerging infectious disease. Okay? Any question about any of that? All right. Let's go ahead and end lesson 1 and 10 and move on to chapter 2. So, as I normally do, the first slide is giving you the major goals and a rough outline. Understand that you need to know more than just the major goals. You need to know all of the objectives, and you can find the objectives on my Canvas web page in the study guide for chapter 2. Occasionally, on some of the lessons, I've got the objectives broken out, and I've got it in a separate link. But if it isn't on a separate link, you'll find it in the study guide. And I'm going to, in the future, have all of my objectives on a separate link, but I'm not there yet. So you need to know the terms, the important terms from chapter 2, and there are more terms, but the most important ones are what's an organic compound, what's an inorganic compound, what's a hydrocarbon. You should know what polar and nonpolar molecules are. You should know the different types of bonds, particularly hydrogen bonds. In reality, you should also know a covalent bond and an ionic bond. You should know what an acid is, a base, and how, what pH is. I'm not going to ask you specifically how we measure pH, but you should know what the pH is. With respect to the 4 major biological molecules, you should be able to describe the relationships and the characteristics of the monomers and the polymers. And before I go further discussing that, can somebody name to me the 4 major biological molecules? You can find them, at least 3 of them, on any cereal box, almost any label. Like this has 1, 2, 3 of them on there. And this is juice, which really only has 1 of them. All the others, I think, are 0. Yep. Nobody knows the 4 biological molecules? The 4 molecules of life? What do you find on a cereal box? Come on, people. Grab a cereal box and start reading it.
So hydrogen,
Carbs, proteins.
Carbohydrates, proteins, that's 2 of them. Hydrogen is not correct, that would be an element. What's 2
Lipids
other
and fats.
biological molecules? We said carbohydrates and protein.
Lipids?
Lipids, which On the label, it won't call it lipid, it'll call it fat, but a fat is a lipid. And then the fourth one isn't on those labels. It would be nucleic acids. Those are the four major biological molecules. So describe the relationship of those four and the characteristics between the monomers and the polymer. The polymer is the big molecule, like the protein is a big molecule, and it's made up of subunits that we call monomers, and each monomer would be an amino acid in the polymer, which is the protein. Be able to describe the composition, the structure, and the function of the four major biological molecules, and then be able to give examples of each. Like for a carbohydrate, you should be able to say something like glucose, fructose, maybe lactose for a disaccharide. Glucose and fructose are monosaccharides. And then starch for a carbohydrate. All right, any question about what we're doing? So in chapter 2, you should know the scale of things. Let me see if I can blow this up. I'm not going to quiz you or test you on the relative sizes of things. That a human is between 1 meter and 10 meter. I think even you guys know that. But I do want you to know that a human is bigger than a chicken egg, which is bigger than a frog egg, which is bigger than most eukaryotic cells, which is bigger than a prokaryotic cell. Oops, went too far. Which is bigger than a virus, which is bigger than a protein, which is bigger than a small molecule like water, which is bigger than atoms. And please note there's an error in this chart. It has 1 micrometer twice. This one is correct. That should be 1 microliter, not 1 milliliter, not 1 microliter right there. So you should know the relative sizes of things, and then anything smaller than a frog egg, we need a microscope to see. And then anything smaller than, let's call it a prokaryotic cell, we need an electron microscope to see. An electron microscope can see larger than that. It can see even a eukaryotic cell. Any question about any of that? So in chapter 2, and we're talking about the biological molecules, it's important to realize what are organic compounds. Organic compounds are typically complex structurally. They have carbon in them, and carbon combined to 4 other atoms. Typically, the molecule is held together by covalent bonds in the carbon. And that's different from inorganic compounds, which typically lack carbon, and typically they're much simpler, not as complex as the organic compounds. But they are important. And inorganic compounds are required for living organisms, for their growth, for their repair, for their maintenance and reproduction. Some examples of an inorganic compound is oxygen, carbon dioxide, and water. Water is one of the most important molecules in a living organism, and it is one of the most abundant, actually it is the most abundant molecule in a living organism. Any question about any of that? So this is getting into your first worksheet. An inorganic compound is water. Water is a polar molecule. What we mean by a polar molecule is that part of the molecule has a partially negative charge, and part of the molecule, the hydrogens, have a partially positive charge. Now, let me state that your textbook calls it a slight charge, not a partial charge. I just want you to realize it's not a full charge. So water does not have a -1 on the oxygen. It is a partially negatively charged atom, and why it is partially negatively charged is the electron sharing between the hydrogen and the oxygen is not equal. The oxygen pulls on those electrons a little bit more than the hydrogen, and so the electron spends more time with the oxygen than with the hydrogen, and that makes the oxygen have a slight negative charge, or like I like to say, a partially negative charge. The oxygen does not have a -1 charge. Okay? The hydrogen, on the other hand, is partially positively charged, and your textbook author would say slightly positively charged, and that's because the electron isn't with the hydrogen as much as it is with the oxygen. The polarity of water allows for hydrogen bonds. What happens is the partially negatively oxygen can be attracted to the positively or partially positively charged hydrogen of another water molecule. And that we call a hydrogen bond, where this partially negatively oxygen is attracted to the partially positively charged hydrogen. So water has two chemical bonds. Between different water molecules, we can have a hydrogen bond. And I should say I'm talking about liquid water, although it would also be true of ice, that you have a hydrogen bond between the water molecules. In steam, you don't have the hydrogen bond. And then water has internal to a water molecule, or within one water molecule, it has covalent bonds linking the hydrogen and the oxygen. So water has two different chemical bonds, within the molecule and between the molecules. Any question on any of that? Water is a good solvent. All polar molecules and ions dissolve in water. What happens is a ion like this, having a negatively charged yellow large molecule and a smaller positive greenly charged molecule, it forms a crystalline structure like that, and the water molecules come up and then surround the positively charged ion, so that the water, this partially negative oxygen, will be showing its face and orienting towards the positively charged sodium ion. And then it can pull that sodium ion out of the crystal and then bring it in solution, and that we'd call the water dissolves the salt, and this would call salt water. And the same thing happens with the negatively charged chloride ion. The water molecule comes up against the negatively charged chloride ion, and the positive hydrogen ions are attracted to the negatively charged chloride ion, and then they come around it and pull it out of the crystalline structure and into solution, with the positively charged, or I should say partially positively charged hydrogen ions oriented towards the negatively charged chloride ion. And this happens with all polar and ionic substances, that water can come in and start dissolving them because of the partially charged structure of water. Any question about any of that? So water is said to be a good solvent. Polar molecules are hydrophilic. What does that mean? Nonpolar molecules are repelled by water. They are hydrophobic. Literally, hydrophobic means fearing water, but what we mean in it is that they do not dissolve in it. Let's come back to here. A polar molecule has a structure like this, where there will be a charge, ionic charge, or a partial charge on a molecule, and then the water will orient itself so that the negative oxygen of water is attracted to the positive, the partially charged molecule of the other one. And if it's an ion, meaning ionic bond, it will be a full charge. And if it is a polar molecule, this will be a partially charged molecule. Non-polar molecules don't have that, like lipids. They will not do this, and lipids will not dissolve in water because they don't have a charged or partially charged molecule. They'll be ionically neutral, and so water can't come in and orient itself to that molecule. I think I went too far. Nope, that's it. Lipids, oil, like mixing oil and water, they'll quickly separate because the oil is nonpolar and the water is polar. Water has a hydrogen bond between water molecules. This makes water an excellent at buffering the temperature, because water can absorb a lot of heat before the water molecule becomes heated and then evaporates. Molecules like oil, which don't have a hydrogen bond between the molecules, the oil molecules can absorb heat, but they can absorb less energy, because when they absorb the heat, the molecules move more rapidly, and then the oil will eventually start to boil when it gets hot enough, and then the oil can't absorb any more heat. Water, on the other hand, can absorb the energy to get the molecules moving more quickly, and that actually is temperature, but we won't get into that. That's one way water molecules absorb energy. But water molecules have a second way of absorbing energy, and that is the hydrogen bond between the water molecules can absorb that energy, and that energy will break the hydrogen bond. And so water has two ways of absorbing energy, and that is why water can absorb a lot of heat, because two things are absorbing this energy. And so water can absorb more energy than most other molecules, like oil. Any question about any of that? Water can split H2O into H plus and OH minus. The H plus is a proton, and the OH minus is called hydroxide ion. It does have a complete negative charge. The charged molecules participate in chemical reactions and give rise to the pH of the water. If there are equal numbers of hydrogen ions and OH minus ions, water will be pH 7. If, on the other hand, there are more H plus ions in solution than OH minus ions, the pH then is negative and less than 7. And if the H plus is less than the OH minus ions, then we have a pH above 7. And that, we say, is increasingly basic. I suppose you should know that wine and tomato juice are acidic, and then grapefruit juice and most other juices are even more acidic. And lemon juice is about the most acidic juice we know, and that's about the same pH as the stomach acid, which can be between 1 and 2. On the other side of pure water, pH 7, we have seawater, which is above pH 8. And then household ammonia is between pH 11 and pH 12. Household bleach is between pH 12 and pH 13. And if anyone uses an oven cleaner, that's the most basic thing you'd probably ever come across, would be your oven cleaner. All right, any question about any of this? pH, I'm not going to hold you to this, is actually the measure of the hydrogen ions in solution. And if you have 10 to the 7th, or is it negative 7th? If you have 1 hydrogen ion and 10 to the 7th water molecules, then you have a pH of 7. When water first breaks apart from H2O to H plus and OH minus, will be pH 7, but because of carbon dioxide being in the air, the carbon dioxide will absorb into the water and then form carbonic acid. We'll talk more about that later. The pH will then drop and it will go to someplace around pH 5. Any question about any of that? So that told us about acids and base. Let's say that an acid is a substance that dissociates in solution, giving up H plus, and that will make the pH less than pH 7. So if we had pure water and then we put HCl in it, the HCl will dissolve in the water and form H plus and Cl minus. The H plus will add to the water, and so we'll have more than a pH 7, and that we call an acid solution, where there's more H pluses than there are OH minuses. A base, on the other hand, is defined by our textbook as giving up OH minus. Like sodium hydroxide dissolved in water will dissociate into sodium ion with the positive charge and OH minus. The OH minus will then get together with the H plus and reduce the H plus in solution, making it less common than pH equal to 7, and that will increase the pH. Any question about any of that? Now you'll note that I mentioned that that's the way your textbook author defines a base. That isn't quite correct. And I'll give you an example. And that is when we put hydrogen carbonate into water, it can absorb H plus in the water, forming carbonic acid, H2CO3, and that will decrease the H plus in the solution. That will raise the pH above 7. And if you don't know, sodium bicarbonate has this, and it is a weak base. So strictly speaking, a base is a substance that gives off OH minus or absorbs H plus ions. And that is why sodium bicarbonate is a base, because it absorbs H plus ions. Okay? And I'm just letting you know that, I don't know, Tortora didn't quite define what a base is correctly. It should be both of these, giving off OH minus or H plus is absorbed. And really, why the OH minus works is this OH minus will combine with the H plus to make water, and that's absorbing the H plus in solution. Okay? Any question about that? All right? If not, a salt is a substance that dissociates in water, forming cations, something with a positive charge, and an anion, something with a negative charge. Sodium chloride, which everybody calls salt, will dissociate into water, sodium chloride, into sodium plus and chloride minus, and so sodium chloride is a salt. But there are other salts, like magnesium chloride. I'm trying to think of another one. Potassium chloride. So those are salts. And when you have salts, meaning ions in water, we call these electrolytes because they allow electricity to flow through the water, meaning electrolytes conduct electricity. And that's why salt water, like ocean water, is more conducive of electricity than, let's say rainwater, for example. Any questions about any of that? I suppose this means if you're in the beach during a thunderstorm where there's lightning, you should probably get out of the water because salt water can conduct electricity very well. But if you were in a freshwater pool, you're probably okay, because freshwater does not conduct electricity. Of course, if they add bleach to the water, the bleach will start conducting the electricity. Organisms need to maintain a fairly constant balance of cellular acids and bases to remain healthy. If they were not to remain, keep their balance of the acids and bases, they would become unhealthy, sick, and actually eventually die. We have a lot of buffers in our body to help maintain our pH. And I've already mentioned the pH is actually the measure of the amount of H plus in solution. Specifically, it's the negative log of the hydrogen ion concentration, and I'm not going to quiz you on that. The pH scale can range from anything between 0 and 14. It's a logarithmic scale. That means when we change the pH by 1, like pH 7 to 6, we're increasing the hydrogen ion concentration tenfold. Any question there? All right, acidic solutions have more hydrogen ions in solution than OH minus ions. Basic or alkaline solutions have more OH minus ions in solution than hydrogen ions in solution. Most organisms grow best around pH 7, so something like 6.5 to 7.5. Most organisms grow best in that pH. And that's true for you too. Your blood is slightly basic. I think it's pH of 7.4. And that's in the normal pH range. All right, any question about any of that? If not, let's come back to the topic of inorganic compounds. Everything shown here is an organic compound. You can see the simplest is methane, CH4. There are more complex here, but this is the most complex one here. And generally, the carbons hold the atoms in organic compound together, meaning all the elements are held by the carbon. And even here, the atoms are held together by the carbon. Of course, the carbon's held together, and the carbon's held together. It's important to realize that there are some links in the molecules of an organic compound which are not held together by the carbon, like these oxygens holding the carbon and the phosphate together. But most of the atoms are held by the carbon. And so carbon is the important backbone of an organic compound, meaning all of these are held together by carbon. We can draw it out this way, but we can also draw it out this way, and this is a molecule which, if you were to draw it like this, each carbon will be where a point is. That would have 2 bonds between 2 carbons. This one would have 1 bond between a carbon, and this is a ring structure. Like I said, wherever there's a point, there's a carbon. So this part here will have 6 carbons, and then the carbons are hooked together there. Any questions about what this is? Oh, let me see. Is there anything else I want to talk about with this slide? Anyways, these are a few examples of some organic chemical compounds. You don't have to memorize that slide. You should just kind of understand what's the difference between an organic compound, like this, and hopefully you know what CO2 and water is. We did show you water earlier. Let's see if I can find it. There's water. And then there's 1 molecule of water, and this is showing you how many water molecules? 5 water molecules with the hydrogen bond being between the water molecules. This is an inorganic compound. This is an organic compound. Or these are organic compounds. Organic compounds always contain carbon and hydrogen. They may contain other elements like oxygen, nitrogen, sulfur, and phosphorus. Carbon and hydrogen in an organic compound are always the main molecules, the main atoms. And all of these elements are held together covalently, and then most of the molecule will be held together covalently by carbon. And that's very different from an inorganic chemical compound. That the organic compounds are generally much more complex. This is much more complex than water, for example. That there is just slightly more complex than water. The complexity of the organic chemical compounds give them complicated biological functions. And let's talk a little bit about the examples of the functions of biological parts of the molecule. So as I mentioned, carbon tends to be held together in an organic chemical compound by the carbon molecules. This is the backbone. But off of the backbone of the carbon, you can have a functional group. And a functional group is some arrangement of atoms that bind to the carbon. And these functional groups give this molecule specific properties, changing the property of the molecule. For example, methane gas, CH4, which I just showed you a little bit right here, is a gas. It doesn't dissolve in water. It does burn very well. When we attach the OH group on it, it changes the property of methane. Methane and methanol, well, methane is a gas, methanol is a liquid. This gives it the ability, the OH functional group gives the methanol the ability to dissolve in water. Methane does not dissolve in water, at least not well. And then this molecule gives this molecule, the functional group gives this molecule polar characteristics because the OH is polar. Any question about that? The important part is functional groups bind to the carbon and they influence the chemical behavior of this molecule because they give the molecule new properties. Like OH gives it the property, instead of being a gas, it's going to be a liquid. It will allow methanol to dissolve in water, among other properties that methane does not have. So the functional groups are responsible for most of the chemical and physical properties of a particular organic chemical compound. And the OH of alcohol is an example, or methanol is an example. Let's take a look at the important functional groups. We mentioned OH, the alcohol group. You can find OHs in lipids and carbohydrates. Another functional group that's important is the amino group, the NH2 group. You can find amino groups in proteins. COOH, and this O has a double bond to the carbon. That O has only a single bond, covalent bond. We call this group, the COOH group, the carboxyl group. You can find this in many of the biological molecules, organic acids, that's a lipid, various lipids, and then proteins and amino acids. And then lastly, the last important and functional group of phosphate, the BOO4, and a phosphate like this has two negative charges. That's a complete charge there and a complete charge there. We find this in phosphate, like phospholipid, and ADP, adenosine triphosphate, as well as DNA and RNA. Why do you think I want you to know phosphate, carboxyl groups, amino groups, and OH groups? And I'm not asking you to know aldehydes, ketones, etc. Why do you think that might be?
Because they're in biological fuels.
Yeah, these are important functional groups in biological molecules, so you need to know them. Okay? Sometimes you can have more than one functional group in a molecule, like an amino acid has at least 2 functional groups. You'll have the amino group on one side of the amino acid and the carboxyl group on the other side. All right, any questions about any of this? If not, I'm going to stop here and I will continue there next time. I'm going to put the 2 here, but all right, I'll see you in the lab at 6:30.
Thank you.
All right?
Thank you.
All right.
Thank you.